Welcome to Nexus Clinical. Before diving into this lecture, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this content is at the user's risk. This is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay seeking medical advice for any medical condition they have, and they should consult their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures. The U.S. Food and Drug Administration's FDA June 2025 decision to remove the Risk Evaluation and Mitigation Strategies (REMS) for 6 approved chimeric antigen receptor (CAR) T-cell therapies marks a significant milestone in regulating advanced biologics. This move indicates a shift from strict, centralized risk management requirements to relying on the mature, decentralized quality systems developed within the clinical community. This change was not sudden deregulation, but the result of years of accumulated clinical experience managing the unique and potentially severe toxicities of these innovative treatments, specifically cytokine release syndrome, CRS, and neurotoxicity. This report will show how the comprehensive, peer-developed standards of the Foundation for the Accreditation of Cellular Therapy, FACT, provided a reliable framework for quality and safety, giving the FDA confidence to reduce burdensome requirements and improve patient access. This analysis will explain the different roles of the FDA Center for Biologics Evaluation and Research, CBER, and Center for Drug Evaluation and Research, CDR, detail the mechanisms behind the previous CAR-T REMS and the strong standards of FACT accreditation, and ultimately examine how establishing quality systems contributed to this historic regulatory change. Based on the content detailing the regulatory oversight of biologics, especially regarding the shift in how chimeric antigen receptor T-cell therapy is regulated, here are 10 brief learning objectives. First, one should be able to distinguish between the regulatory roles of the Center for Biologics Evaluation and Research and the Center for Drug Evaluation and Research by understanding their focus on product complexity. It is also important to identify cell and gene therapies as products primarily overseen by the Office of Therapeutic Products within the Center for Biologics Evaluation and Research. Furthermore, one should recognize the scientific risks associated with living drugs such as cell and gene therapies, which include insertional mutagenesis and toxicities related to vectors used in their creation. Explaining the purpose of risk evaluation and mitigation strategies is also essential. Specifically, understanding that these strategies are established by law to manage serious toxicities, exemplified by the design of the chimeric antigen receptor T-cell therapy risk mitigation plan to address cytokine release syndrome and neurotoxicity, also known as immune effector cell-associated neurotoxicity syndrome. Detailing the historical requirements of these risk mitigation strategies is necessary, including mandates for on-site access to the drug tocilizumab, and a 4-week proximity rule for patients. Analyzing how these requirements created logistical and financial burdens helps to understand their impact on patient access to care. It is important to identify the Foundation for the Accreditation of Cellular Therapy as an organization with the mission to develop evidence-based quality standards through peer review. Its core principle involves establishing and maintaining a comprehensive quality management program. Explaining why the Food and Drug Administration decided to eliminate the risk mitigation strategies in June 2025 reveals that this decision was influenced by the increased maturity of the clinical community and greater reliance on institutionalized quality systems, such as those developed by the Foundation for the Accreditation of Cellular Therapy. Finally, despite the elimination of these risk mitigation strategies, certain safety measures remain mandatory. In particular, it is vital to state that manufacturers of cell and gene therapies are now required to conduct a post-marketing observational safety study extending for 15 years to continually monitor safety after the product reaches the market. Biological products, or biologics, are a diverse group of therapies made from living organisms, including humans, animals, and microorganisms. In the United States, their regulation is mainly governed by the Public Health Service, PHS, Act and the Federal Food, Drug, and Cosmetic, FD&C Act. Unlike chemically made small molecule drugs, which have a clear structure, biologics are often large, complex molecules or mixtures of molecules. Many, such as cell and gene therapies, are considered living drugs capable of persisting, replicating, or integrating within the patient. This inherent complexity and variability require a specialized regulatory approach to ensure their safety, purity, and potency. Consequently, FDA oversight reflects the unique scientific challenges these products pose. The Center for Biologics Evaluation and Research, CBR, is the primary entity within the FDA responsible for regulating the most complex biological products. Its mission is to protect and advance public health by ensuring that biologics are safe, effective, and available to those who need them. The Center for Biologics Evaluation and Research has a broad scope of authority that includes a wide variety of products. These products encompass vaccines used to prevent infectious diseases, as well as blood and blood components intended for transfusion. Additionally, it oversees pharmaceuticals derived from blood, such as clotting factors. Human tissues and cellular products employed in transplantation, including stem cells, are also within its jurisdiction. The agency regulates allergenic extracts and diagnostic patch tests as well. Furthermore, it covers products involved in xenotransplantation, which use live animal cells or tissues. Importantly, the center also governs cell and gene therapy products. These therapies aim to modify or manipulate gene expression to treat various conditions. CBR's regulatory authority is comprehensive, extending from the pre-market review of investigational products to robust post-market surveillance. This includes inspecting manufacturing facilities before and after product approval, conducting confirmatory lot release testing for certain products like viral vaccines, and monitoring adverse event reports through systems such as MedWatch and the Vaccine Adverse Event Reporting System, VAERS. The Center for Biologics Evaluation and Research handles the most complex biological products. Meanwhile, the Center for Drug Evaluation and Research oversees the majority of traditional small molecule drugs. It also manages a significant portion of the biologics portfolio, particularly those that are less complex and better understood. This includes therapeutic proteins such as monoclonal antibodies, cytokines like interferons, therapeutic enzymes, and certain immunomodulators designed to modify immune responses. A major organizational change took place in 2002 when the Food and Drug Administration transferred oversight of many therapeutic proteins from the Center for Biologics Evaluation and Research to the Center for Drug Evaluation and Research. This move consolidated the review of these products within a center more familiar with traditional pharmaceutical development. The review process for biologics in this center, usually submitted through a biologics license application, emphasizes chemistry, manufacturing, and controls, as well as pharmacology and clinical trial data. This approach closely mirrors how the center reviews new drug applications. The division of labor between CBR and CDR is more than just bureaucratic. It's a strategic organizational design grounded in the fundamental scientific nature of the products they regulate. The key difference is product complexity. CBR's portfolio mainly includes products that are living entities, cells, tissues, derived from complex biological systems, blood products, or intended to provoke complex immune responses, vaccines. In contrast, CDR handles purified, well-understood proteins and peptides that, although biological in origin, act more predictably from a manufacturing and pharmacological perspective. This jurisdictional separation reflects a deeper scientific and philosophical divide. The unique risks linked to CBR-regulated products, such as persistence, replication, host genome integration, and unpredictable immune responses from living drugs, necessitate a different set of scientific expertise than what's needed for the pharmacokinetic and pharmacodynamic analysis of a monoclonal antibody. The FDA has thus structured itself to incorporate specialized knowledge in virology, cell biology, gene therapy, and immunology within CBER to oversee the products that require it most. For developers of new therapies, understanding this division is a crucial first step. Within CBER, the Office of Therapeutic Products, OTP, has been created as the dedicated division responsible for reviewing and overseeing CGTs, bringing together the expertise needed to manage this rapidly evolving field. The CBER Product Jurisdiction Officer serves as a vital resource for sponsors to get a formal decision on which center will regulate their product, a decision that guides all subsequent regulatory, clinical, and manufacturing strategies. This initial classification is one of the most important early steps in product development, as it determines which set of strict standards and guidance documents, such as CBER's requirement for 15-year long-term follow-up for gene therapies, will influence years of work and significant financial investment. Cell and gene therapies mark a major shift in medicine, aiming to treat or prevent diseases by adding, removing, or changing genetic material within a patient's cells. This transformative potential comes with a unique and complex risk profile that requires a specialized regulatory system. Unlike traditional medicines that are made in large, uniform batches, many CGTs are patient-specific, autologous, use viral vectors for genetic modification, and are designed to stay in the body for years or even a lifetime. These features introduce new risks not seen with conventional drugs. These include the chance of insertional mutagenesis, where the therapeutic gene inserts into the host genome and disrupts an important gene, immune responses against the vector or the therapeutic protein, vector-related side effects, and the potential for delayed adverse events that may only appear years later. The manufacturing process itself adds complexity, especially for ex vivo therapies like CAR-T cells, which involve taking a patient's cells, modifying them in a lab, and reinfusing them, a multi-step process that needs strict control. This is different from in vivo therapies, where the genetic material is delivered directly into the patient, creating different challenges related to targeting and immune reactions. Recognizing the specialized expertise required to oversee this field, the Center for Biologics Evaluation and Research has consolidated its cell and gene therapy review functions within the Office of Therapeutic Products. The establishment and recent reorganization of this office have led to a doubling of its approval rate for new cell and gene therapies in 2024. This development underscores the Food and Drug Administration's commitment to building and maintaining a dedicated team of experts to manage the growing pipeline of these products. The role of the Office of Therapeutic Products extends beyond mere review and approval. It is designed to be a proactive partner for sponsors. The Center for Biologics Evaluation and Research provides scientific and regulatory advice to researchers and manufacturers from the earliest stages of development. This collaborative approach is essential for navigating the novel scientific and regulatory challenges posed by cell and gene therapies. This commitment to efficiency and support is further reflected in the agency's commitments under the Prescription Drug User Fee Act, which include measures to streamline and support the development of cell and gene therapy products. To provide clarity in this complex area, CBER has developed and continuously updates an extensive library of guidance documents tailored specifically to CGT products. This body of guidance demonstrates an adaptive and precautionary regulatory model. It is adaptive in that it responds directly to emerging scientific advancements, with specific guidance on topics like human genome editing, CAR-T cell development, and studying multiple product versions in early trials. It is precautionary in its recognition of the long-term uncertainties associated with these durable therapies. The guidance provided by the Center for Biologics Evaluation and Research addresses several key areas. It includes detailed recommendations for chemistry, manufacturing, and control processes, focusing on product manufacturing and characterization. The guidance also emphasizes the importance of potency assurance by outlining expectations for developing assays that verify each product lot has the necessary biological activity. Additionally, it offers guidance for managing manufacturing changes and maintaining product comparability, ensuring that process modifications do not compromise safety or efficacy. Long-term follow-up is another critical aspect, requiring extended monitoring of patients after treatment. For gene therapy products, this follow-up is especially important and can last up to 15 years to monitor for delayed adverse events, such as the potential development of secondary cancers. This intense focus on manufacturing highlights a key principle in the regulation of autologous therapies. The process is the product. Since each dose is made for a single patient, the consistency, control, and validation of the entire manufacturing process are as important to the final product's safety and effectiveness as its release criteria. CBER's detailed guidance on CMC and comparability is not just about quality control in a traditional sense. It aims to ensure the integrity of the therapeutic product provided to each patient. The FDA's oversight of CGTs continues even after approval. Post-market surveillance is highly rigorous to track the long-term effects of these therapies. Manufacturers must promptly report adverse events and product deviations to the FDA. Importantly, even if other post-market requirements are changed, the essential obligation for long-term safety monitoring stays in place. As shown by the recent removal of REMS for certain CAR-T products, the FDA clearly stated that manufacturers must carry out 15-year post-marketing observational safety studies. This ongoing effort guarantees that the long-term safety of these therapies, including the risk of secondary cancers, is continuously monitored well after they are introduced to the market. A risk evaluation and mitigation strategy, REMS, is a formal safety program that the FDA can require for certain medications with serious safety concerns. Its statutory purpose is to ensure that the benefits of a medication continue to outweigh its known risks. REMS are not a standard part of drug regulation. They are reserved for products where standard professional labeling is considered insufficient to manage a specific, significant risk. The elements of a REMS can range from a simple medication guide for patients to more complex and restrictive requirements known as elements to assure safe use, EDISU, which may include provider certification, patient monitoring, or dispensing in specific healthcare settings. Since their initial approval in 2017, all commercial CAR-T cell therapies have been subject to a risk evaluation and mitigation strategy, REMS, with elemental, targeted, and accessory services, EDISU. This requirement was implemented in response to the well-known, serious, and potentially life-threatening risks of cytokine release syndrome, CRS, and immune effector cell-associated neurotoxicity syndrome, ICANS. CRS is a systemic inflammatory response caused by the extensive activation of CAR-T cells, while ICANS is a neurological toxicity with a variety of symptoms. The REMS program was created as a controlled system to ensure these potent therapies are only given in healthcare settings equipped to recognize and manage these complex and acute toxicities. Initially, with clinical experience mainly limited to a few specialized academic centers, the FDA implemented the REMS as a centralized, prescriptive tool to ensure a baseline of safety and institutional readiness across all potential treatment locations. It was a blunt but necessary instrument to manage risk during the initial rollout of a transformative yet potentially hazardous technology. The historical CAR-T REMS programs imposed specific and often burdensome requirements on both healthcare providers and patients. These elements were designed to create a controlled environment for therapy administration and post-infusion monitoring. For healthcare providers and facilities, hospitals and their associated clinics had to obtain special certification for each individual chimeric antigen receptor T-cell therapy product they intended to administer. A key component of this certification process was the requirement to have immediate access on-site to tocilizumab, an interleukin-6 receptor antagonist. This medication is a critical treatment for severe cytokine release syndrome, a serious potential side effect. Ensuring the availability of tocilizumab helped guarantee that the primary remedy for the most common life-threatening toxicity was quickly accessible when needed. For patients receiving treatment, the risk evaluation and mitigation strategy placed significant logistical, financial, and personal burdens on both patients and their caregivers. One of the key requirements was proximity monitoring, which required patients to stay close to the certified treatment center. often within a 30-minute to 2-hour drive, for at least 4 weeks following the infusion of chimeric antigen receptor T cell therapy. Additionally, patients were advised to avoid driving or operating heavy machinery for 8 weeks after treatment due to the risk of delayed neurotoxicity, a potential serious side effect. These precautions were designed to ensure safety, but added considerable challenges for patients during their recovery period. The burdens and barriers of REMS. Impact on patient access and the healthcare system. Although medically justified when implemented, the REMS requirements revealed a fundamental conflict between prioritizing safety and facilitating access. The patient-facing components, in particular, created significant real-world challenges. The 4-week proximity rule often compelled patients and their families to find and cover the costs of extended temporary lodging near treatment centers, posing a major obstacle for those from rural, remote, or economically disadvantaged backgrounds. These non-medical burdens, including lost wages, caregiver stress, and housing expenses, effectively formed socioeconomic barriers to care, potentially excluding patients who were clinically eligible but unable to handle the logistical demands. For healthcare institutions, the product-specific certification process, training requirements, and other administrative tasks added complexity and drained resources, which could limit the number of centers able to provide these therapies. The FDA later acknowledged that a key goal of eliminating the REMS was to minimize the burden on the healthcare delivery system. Under the historical framework of risk evaluation and mitigation strategies, provider and facility certification was necessary for each specific chimeric antigen receptor T-cell therapy product. This certification requirement has been eliminated in the current framework, following the end of the Risk Evaluation and Mitigation Strategies program in June 2025. Similarly, on-site, immediate access to tocilizumab was previously mandated before infusion, but this requirement is no longer in effect. Regarding patient proximity, patients were previously required to remain near the healthcare facility for at least 4 weeks after infusion. Currently, this recommendation has been reduced to an advisory of 2 weeks after infusion. The restriction on patients driving themselves has also been lessened, from a recommendation of 8 weeks to just 2 weeks after infusion. Patient monitoring practices have also evolved. Previously, patients were monitored daily for approximately 4 weeks following infusion. The updated protocol now recommends daily monitoring for at least 1 week, with continued observation for at least 2 weeks after infusion. Communication about risks, which was formerly conveyed through materials associated with the Risk Evaluation and Mitigation Strategies program, boxed warnings, and medication guides, is now primarily communicated via the boxed warning and medication guide. Long-term follow-up remains consistent, requiring a 15-year post-marketing observational safety study. The Foundation for the Accreditation of Cellular Therapy, FACT, is a nonprofit organization founded in 1996 through a partnership between the International Society for Cellular Therapy, ISCT, and the American Society for Transplantation and Cellular Therapy, STCT. Its main goal is to promote quality medical and laboratory practices across the full range of cellular therapy. It accomplishes this by establishing strict, evidence-based standards and providing a voluntary accreditation program that ensures compliance with these standards. These standards are not set by regulators, but are developed through a consensus process by international teams of leading experts, clinicians, and scientists actively engaged in the field. Consequently, FACT standards embody the core clinical best practices, a grassroots articulation of what top practitioners deem essential for a high-quality, safe cellular therapy program. The foundational principle of FACT is that quality patient care can only be achieved if all aspects of the therapeutic process, from clinical management and donor selection to cell collection, processing, and administration, are effectively addressed and integrated within a single, overarching quality framework. The cornerstone of the FACT standards is the requirement for every accredited program to establish and maintain a comprehensive quality management (QM) program. This system is more than just a simple checklist. It is a dynamic and integrated framework that encompasses several critical components. Firstly, it involves training personnel and assessing their competencies to ensure they are qualified for their roles. Additionally, It requires the development and maintenance of written policies and standard operating procedures for all essential activities. Furthermore, the system includes the implementation of validated processes to guarantee consistency and effective control. A key element of this system is the establishment of robust procedures for managing occurrences. This includes tracking and thoroughly investigating adverse events, errors, accidents, and complaints. The system also mandates the implementation of corrective and preventive actions to address identified issues and prevent future occurrences. Recognizing that therapies like CAR T cells present unique challenges, FACT developed a specific set of standards for immune effector cells, IECs, first published in 2017. These standards apply to a range of products, including CAR T cells, natural killer, NK, cells, dendritic cells, and therapeutic vaccines. The IEC standards are comprehensive and establish a broad, quality ecosystem that goes well beyond the limited scope of a REMS. While a REMS targets a few specific, high-profile risks, FACT accreditation requires a holistic quality system that covers every aspect of the therapeutic process. This system offers a much more thorough and proactive assurance of quality and patient safety. Effective organizational management is essential, requiring an integrated team with clearly defined leadership responsibilities and accountability. This structure helps ensure compliance with regulatory standards and maintains the continuity of care. Personnel and training standards are stringent. Program directors and staff must meet specific educational and experiential qualifications. Additionally, ongoing training and competency assessments are mandatory for all personnel to maintain high standards. A comprehensive and integrated quality management plan is necessary. Such a plan should encompass process control measures, data collection protocols, document management systems, and strategies for continuous improvement. These improvements are achieved through regular internal audits and the implementation of corrective and preventive actions. Clinical operations must be supported by facilities that are adequate and equipped to provide emergency and intensive care services. It is vital to have access to critical pharmaceutical agents to manage toxicities effectively. Moreover, robust protocols are required to guide patient selection, administration procedures, and the management of adverse events. Facilities responsible for cell collection, processing, storage, and transportation must adhere to detailed standards. These standards are designed to preserve product integrity, sterility, and viability throughout the entire process. Lastly, procedures for product labeling and maintaining the chain of identity are strictly enforced. Proper coding and labeling ensure traceability and help prevent errors. It is also essential to maintain a complete chain of identity and custody, from the donor or patient to the final administration of the product. Achieving FACT accreditation is a rigorous process that provides strong external validation of a program's dedication to quality. It starts with submitting comprehensive documentation showing compliance with all relevant standards. This is followed by a multi-day on-site inspection by a team of trained, peer experts who review records, observe procedures, interview staff, and confirm that the program's written SOPs are being followed in practice. Accreditation is awarded for a 3-year cycle, during which the program must maintain compliance and demonstrate ongoing quality improvement efforts. This thorough process ensures that FACT accreditation is not just a one-time achievement, but a continuous commitment to excellence. The FDA's June 2025 decision to remove the REMS requirement for all 6 approved BCMA and CD19 directed autologous CAR T-cell therapies did not happen in isolation. It resulted from a collaborative effort by the clinical community to show that the field had advanced enough to make the REMS unnecessary. A key event was a June 2023 workshop organized by the ASTCT, which gathered over 70 stakeholders, including clinicians, regulators, manufacturers, and representatives from accrediting organizations like FACT. The main consensus from this meeting was that the existing clinical infrastructure, based on professional society guidelines, transparent data reporting to registries, and strong accreditation standards, was enough to ensure safe use of these therapies without an additional REMS. These conclusions were formally presented to the FDA and published in a peer-reviewed journal, offering a solid, evidence-based case for changing the policy. The FDA's official rationale for the decision directly mirrored the arguments made by the clinical community. The agency stated that a REMS was no longer necessary to ensure that the benefits of these CAR T-cell immunotherapies outweigh their risks. The justification was explicitly based on the established management guidelines and extensive experience of the medical hematology/oncology community in diagnosing and managing the risks of CRS and neurologic toxicities. Top FDA officials reinforced this message. Dr. Richard Pazder, director of the Oncology Center of Excellence, noted, "Physicians and institutions now have greater experience identifying and managing toxicities," while Dr. Vinay Prasad, director of CBR, framed the reevaluation as a core part of good regulatory practice, stating that eliminating a REMS that is no longer needed expedites the delivery of potentially curative treatments to patients and reduces burden on providers. The decision was further supported by data showing that adverse event reporting for CRS and neurotoxicity had remained stable over time, indicating that the risks were being managed effectively under existing clinical practice. The FDA's confidence in the established management guidelines of the clinical community is, in large part, a reflection of the widespread adoption of comprehensive quality systems like those mandated by FACT. A direct comparison reveals that the robust, integrated requirements of FACT accreditation meet or exceed the safety objectives of the more narrowly focused REMS. The objective of the risk evaluation and mitigation strategy is to ensure site readiness and effective management of toxicity related to treatment. To achieve this, sites are required to undergo certification and gain on-site access to tocilizumab, a medication used to manage cytokine release syndrome. This preparation is essential for ensuring the facility's capability to handle potential adverse reactions. In comparison, the standards set by the Foundation for the Accreditation of Cellular Therapy offer a more comprehensive approach. These standards mandate that accredited clinical programs have established procedures for emergency and intensive care. Additionally, they must ensure access to all necessary pharmaceutical agents required for managing toxicity. Accredited programs are also required to develop detailed standard operating procedures and have trained personnel specifically equipped to identify and manage the full range of complications that can occur after infusion. A site that is accredited by the Foundation for the Accreditation of Cellular Therapy has demonstrated institutional readiness through a thorough and independent peer review process, confirming its capability to provide safe and effective treatment. The risk evaluation and mitigation strategy aimed to ensure that healthcare providers were well educated about the specific risks associated with cytokine release syndrome and immune effector cell-associated neurotoxicity syndrome. These are serious potential toxicities that require thorough understanding and management. The standards established by the Foundation for the Accreditation of Cellular Therapy address this need by implementing more extensive requirements for personnel qualifications. These requirements include specific education, training, and experience related to cellular therapy. Additionally, the Foundation mandates ongoing assessments of competency to ensure that all clinical staff remain current in managing these complex patients and their potential toxicities. The objective of the Risk Evaluation and Mitigation Strategies (REMS) program was to establish a uniform, top-down set of rules applicable to all certified centers. However, the accreditation process by the Facility Accreditation Council for Testing (FACT) offers a more comprehensive and organic form of standardization. FACT accreditation requires each program to develop, validate, and follow a detailed set of internal standard operating procedures that are integrated into a facility-wide quality management program. This system, which includes regular audits and corrective action plans, fosters a deeply embedded and self-sustaining culture of quality and process control. The removal of the CAR-T REMS is a notable example of adaptive regulation and showcases the strength of professional self-regulation. The FDA initially employed a prescriptive, centralized approach, REMS, when the field was still developing and experience was limited. As the clinical community grew, it established its own strong, decentralized quality assurance systems, which were formalized and validated through the FACT accreditation program. The FDA's decision indicates its acknowledgement that these institutionalized quality systems had effectively replaced the need for the original federal mandate. This marks a significant shift from the FDA enforcing a safety system to the FDA recognizing and trusting a safety system created by the clinical community itself. This regulatory development creates a positive cycle that encourages quality. By reducing a major administrative and logistical burden for institutions that have already invested in high-quality infrastructure, often shown through FACT accreditation, the FDA implicitly rewards that investment. This indicates that committing to a comprehensive quality system is not only beneficial for patient safety, but can also lead to a more efficient and less burdensome regulatory process. This example may promote broader adoption of FACT accreditation for future innovative therapies, fostering a cooperative oversight model based on mutual trust between regulators and the regulated community. The most immediate and tangible effect of removing the REMS is a significant decrease in barriers to patient care. Reducing the required patient proximity period from 4 weeks to 2, and the driving restriction from 8 weeks to 2, will greatly lessen the logistical and financial burdens on patients and their families. This change is especially beneficial for those living in rural or underserved areas, where the previous rules often made treatment extremely difficult. For providers and healthcare systems, eliminating the product-specific site certification and other administrative requirements lowers complexity and resource use, potentially enabling more centers to provide CAR-T therapy and helping to expand access to these life-saving treatments. It is essential to understand that removing the REMS does not mean safety oversight is relaxed. The FDA has clearly stated that critical risk information will still be communicated to providers and patients through the product's prominent boxed warning for CRS and neurological toxicities, as well as the detailed patient medication guide. Additionally, the most important long-term safety measure remains in place: the requirement for manufacturers to conduct 15-year post-marketing observational safety studies. This mandate ensures that the long-term risk profile of these therapies, including the potential for secondary malignancies, will be carefully and continuously monitored for more than a decade after use. The journey of CAR-T regulation can serve as a model for future advanced therapies. It indicates a possible regulatory life cycle where a new, high-risk therapy might begin with a restrictive REMS to control its rollout. However, as clinical experience grows and reliable, verifiable quality systems like those defined by FACT become standard practice, the FDA may shift away from strict initial requirements in favor of professional standards and ongoing post-market surveillance. This flexible approach allows the FDA to remain cautious at the start of a new technology while also being adaptable enough to ease burdens and enhance access as the field develops. The evolution of CAR-T regulation from a mandatory REMS to relying on institutionalized quality systems signifies the maturing of not just a therapeutic approach, but also the entire clinical and regulatory framework that supports it. It powerfully illustrates successful collaboration among regulators, the biopharmaceutical industry, and the clinical community. By acknowledging the strong, peer-developed standards that now support the safe use of cellular therapies, the FDA has approved a new, more efficient, and sustainable oversight model. This paradigm shift, grounded in proven clinical competence and comprehensive quality management, opens the way for revolutionary therapies to be delivered safely, effectively, and with broader access for all in need. This was an episode of the Nexus Clinical Podcast. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this content is at the user's risk. This is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay seeking medical advice for any medical condition they have, and they should consult their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.